So I'm Itsik from Columbia University, from the Department of Computer Science, and also the Department of Systems Biology, and I'm going to be giving a tutorial about variational inference in population genetics, at which point you might have questions about, having been here yesterday afternoon for David Noll's talk about variational inference, how come you're having two faculty from the same department giving tutorials about essentially the same computational technique. And there are several good reasons for that. First of all, variational inference is very cool and a very powerful set of methods that allow us to do things that we weren't able to just a couple of years back. So no wonder at our institution and elsewhere there emerged critical masses of people who are excited about these tools and developing them and applying them across genomics and beyond. The second reason is that while very awesome, some equations are love at first sight, some requires some more reiteration and seeing them multiple times in order to actually realize how to apply them across different contexts and different types of problems. So we thought it would be useful to showcase another context. So I'm sure you all rushed off after David's talk to implement stochastic gradient descent, but I'm also sure you only reached a local maximum at Castle Rock. So, and last but not least, a third reason is that if you'd noticed the schedule over the last few weeks, it had changed due to some change of plans by some faculty. So Zara asked me to do this back to back. But going forward, what we're going to be doing is first reviewing or representing variational inference and how and why, and getting to the nitty-gritty of really equations and excruciating detail, and somewhat opposite to the classical order of things, would then go to the actual motivation that leads us or led us to this area, and engaging in some more research that builds off a more fundamental example that we're going to use for the first part, and the research work, as well as some of the slides, is all due to my wonderful student Tyler Joseph, and I wanted to make sure that I credit him right off the bat. Do stop me at any point if there are particular questions, because the goal is for this to be an interactive tutorial. All right, so variational inference is a broadly applied technique, but in population genetics, perhaps a classical example for this has to do with understanding the ancestral composition of individuals. So who here has their genome done in 23andMe or MyHeritage or any other company or all of the above? Right, at least some of you. And so each of you might be a vertical line in this spot. If you have access to your genome, you might have seen the analysis that all these companies, as well as many research papers that deal with genetic data, provide, which is analysis of ancestry components of different individuals. In this example, different colors, mostly mostly brown, pink, and green here, correspond to different populations of origin, say Sub-Saharan African, East Asian, European, and while this individual, this column of pixel might correspond to an individual with only pink ancestry, this individual has mixed ancestry, some proportion of the of green ancestry and mostly pink. This has been a staple of genetic analysis of the last couple of decades, ever since Pritchard, Stephens, and Donnelly introduces a program and algorithm called structure to analyze population structure. The input is just the genotype of me, you, and everybody else in different rows across many loci. Maybe a few loci when this had been introduced. This was pre-whole genome genotyping, but nowadays, obviously, we can ascertain the entire genome and get variants everywhere. Now, if you try to formalize this and think of this as a computation, so who here had seen papers, worked with papers that consider this analysis? A couple. Okay, all right. So, but is there a general idea? Does it generally make sense? Right. So let's try to see what's the formal model for that and how variational inference can be useful here, because when this was introduced, not only did whole genome genotyping not exist, but also variational inference was not yet developed. So doing this analysis involved lots and lots of CPU running very intensive Monte Carlo Markov chain simulations in order to converge to this kind of a model. So formally, what is this model? And this is following the plate model notation that David introduced. So if you're trying to generate the genotypes in through a probabilistic model, what structure says one would need to do is first, for each of the K populations and for each of the L loci, we need to decide on allele frequencies for that particular population, for that particular locus. So we draw betas, beta KL, which is just an allele frequency for that SNP for that particular population. Now, we also need to go through each and every individual, and for each individual, draw its ancestry proportions, the kind of pie chart or vector of pixels that you get from direct-to-consumer companies, and we'll call that theta D for each individual D. Again, the fact that it's within a plate means that we're going to do that for each small d in the set of capital D individuals. Having done that, we can go over that individual's genome locus by locus, and for each locus we can draw the population that this locus is coming from, a variable that we call ZDL, that's for each individual D for each locus L. And if we know the population of origin and we know the allele frequencies at that locus for that population of origin, we can actually flip coins and draw the actual genotype that exists for that individual at that locus. And that's the only thing that we actually observe as data. So that's questions about the plate model. All right, then we can actually write that down as a formal probabilistic model and realize what else might be involved here. So we draw the allele frequencies beta KL, not very originally, but from a beta distribution with some fixed parameters. We draw the individual proportions, individual ancestry proportions for each individual, theta d, with Dirichlet prior, with some parameters that we might assume there. And if we already have the ancestry proportion, so if I know that I am 30% blue, 20% green, and 50% red, then I can now draw my ancestry at each locus using these proportions. So each ZDL is a discrete variable that says blue, red, or green, and with these particular proportions for me or for each individual D, just using this discrete distribution. Once we have the population for ancestry for me at each locus, I can draw a genotype at that locus for me using the allele frequencies for that population. So if I drew a particular SNP to come from the red population, then I'm going to use beta for the red population when I'm drawing my genotypes at that locus, and we're going to do that using the binomial distribution. We're just flipping 2 coins to draw these alleles. Okay. At the end of the day, we're going to have our wonderful matrix X of genotypes across loci and across individuals. Okay, so this is just this is the endpoint of the simulation in a generative model. This is just the starting point for our inference problem. We just see this, and we want to infer everything else. So we observe the genotype data, X for each individual, D for each local cell, and we want to estimate the allele frequencies for the different populations of different loci, the admixture proportions for each individual, and also along the way figure out the unknown, the hidden assignments of ancestry for each locus along the genome. So how do we do that? Well, Bayes' rule tells us that we would like to figure out the probability for a particular estimated set of parameters given what we observe. Right? So we need to compute the probability for betas and thetas and z's given the observed data X. Right? How do you compute conditional probability? So Bayes' rule has been there for a couple of centuries, and we can just compute the probability of everything. So for each K and for each L, we just multiply the probability of drawing the betas, because we don't rely on anything in order to draw them. We multiply all that by the probability of the thetas that we draw, because again, they don't rely on anything external. And then after we did that, well, we need to go over locus by locus, and within this nested loop, we need to also count in the probability of z given theta, and then x given z and beta. Just following up all through the the generative model and computing the full likelihood of beta, theta, z, and x. And since it's conditional, we need to divide it by just the probability of x. So far, so good? Yeah.
So the way you've written it right here with the beta and the thetas separated out and multiplying, that's assuming that your allele frequencies and admixture proportion is your allele frequencies and admixture proportions, the betas and the
yes. To the yeah.
Here you go.
Yes.
Yeah. So you're multiplying the betas and the thetas. Doesn't the allele frequencies depend on the admixture proportions?
Yes. So the nested loop you should do that in a nested loop fashion. Mm-hmm. You're right. Yeah. We should do that for each. Perhaps the best way to do it. So the right way to do that would to write a parenthesis structure that's not here. You're right.
Is the question whether the prior of the allele frequency should depend on the admixture proportions, or another question?
Yeah, in the numerator, right?
In the numerator.
This guy and this guy separated out right here. Does this mean that those are independent from each other?
Yeah. So yes, they are. So okay, well let's go to the model. So we're drawing the allele frequencies separately. We don't need anything in order to do that. And we're drawing the answer proportions separately. So that's the assumption of how the data had been generated. Okay.
Confusing in this model is that you might think that the data depend on how diverse the populations are, and there would be a correlation, and that model doesn't recognize that. So that might be the confusing part. Mm-hmm. But this data is what happened after these populations diverged when the populations got in contact recently again. So these are completely independent. So you imagine you have these different populations with randomly drawn allele frequencies, and then they are brought together at random. Got it. Okay.
So the model is somewhat rudimentary, and in fact, maybe its simplicity explains why it has been so successful. And perhaps it's surprising how successful it had been, given how simple it is. But it does assume that different individuals have answer proportions that are individually drawn without any relation to one another, and that the allele frequencies for each particular variant in each particular population are just drawn independently. This is part of the critique that exists against this model, and in fact, if we have time and go into applications, we'll actually discuss how one can address that also with variational inference. Okay. Well, okay. So the real issue comes up when we need to actually address the denominator, because the denominator tries to evaluate the probability of the data. We're trying to estimate the conditional probability of the model given the data, and the probability of the data can be computed by integrating over all the parameters we don't know and evaluating this complete probability, this complete likelihood at each particular point in this space. Now, again, one can do that or can approximate that or estimate that through MCMC or related methods, but the big picture message is that integration is hard, and we want to avoid that if we can. Fortunately, we have a way out. And the big idea is that instead of working with a posterior distribution that we really don't like because it involves computing ugly integrals, we will replace that distribution with a different distribution. We'll replace this P by a Q that would come up with in a couple of minutes, less than 14. And that distribution would be a distribution of the same parameters of the model, those betas and the thetas and the z's. It is most conveniently distribution from some family of distributions. So it's parameterized by some parameters of that family. So we'll call those parameters eta. And we'll choose Q in a way that's convenient to us. So so we'd be able to conduct inference within Q as our distribution. So I was sort of suggesting that would choose an arbitrary Q that, though would not make sense, would like this Q to approximate the actual distribution that we're interested in. So we're gonna choose a distribution Q within this family by choosing a particular set of parameters, eta. We'll choose eta hat, a set of parameters that would make Q as similar as we can to the ugly distribution P. So we're going to minimize the distance between these two distributions. How do we measure distance between distributions? KL divergence, right? So we'll choose eta that minimizes the KL divergence between distribution Q that we're suggesting, Q of all these parameters that we'll call Y in order to fit on the slide, and the conditional distribution of Y given the actual data. And you may recall that the KL divergence is just the integration of the log Q over P dq, so weighted by this distribution Q, and it's a distance measure. It's always positive. Okay. Now, so we took something that's intractable, and we're trying to replace that with a distribution that's tractable and would make life easy for us. And we're going to write this. Does this help us? So it would be great if it only included Q, but we didn't get rid of P. We still have P here. So what have we done with all this notation? Well, here there's a standard manipulation that is useful to be familiar with that comes to our rescue. So if we take just the overall log likelihood, log of P of X, and expand that by integrating dq, so we write down log P of X as the integral of log P of X times Q of Y dy, then we can, of course, multiply and divide by the same number. We haven't done anything yet, but that allows us to partition this expression and write P of X as the ratio between the complete likelihood of X and Y divided by the conditional likelihood, the probability of Y given X, and separating out the two terms of the log. It's log of a product, so these are two things that are added up and separate to two separate integrals. We get two terms. We get log of P of xy divided by Q of y times Q of y dy, and log of Q of y divided by the posterior probability of y given x times Q of y dy. Now, does any of these terms look familiar? The right one is the KL divergence that we just saw, right? And since it's a KL divergence, we know it's non-negative, which means that the left part that can be rewritten as the expectation according to Q of this expression of the log conditional likelihood. This expression is now a lower bound on the overall log likelihood. So this is typically called the evidence lower bound or elbow, if you run into that expression. And this is now great because while we don't want to compute the KL divergence directly because it involves the ugly distribution, we don't want to deal with the posterior distribution that involves integration. We now have a nicer expression that's a lower bound on the overall log likelihood that does not involve any integration over the probability that we don't want to integrate. Okay, so that's really the main idea. And now, instead of finding the distribution Q that minimizes the KL divergence, so that's more similar to the distribution P that we're after, we can just maximize the elbow, maximize the other term that sum up with the KL divergence to the same thing. Okay, so instead of finding the minimum eta for KL divergence, we just maximize the elbow. Right? At this stage, variational inference sort of have completed the transformation of integration that we don't like to an optimization problem that where we have a whole battery of tools available to us and can apply them. And now there are two options. If you have lots of Google Cloud credits on the grant that you have to spend and don't want to think about it too much, well, one can come up with any kind of family of distributions Q that's convenient to work with and apply direct optimization to this methodology. TensorFlow had really revolutionized our ability to follow up on such models and do that with not too much programming involved. But the downside is that this may or may not work as well, and a more thoughtful approach would be to use mean field theory and follow up variational inference by optimizing one coordinate at a time, which I have 0 minutes to talk about. Couple. Okay. 3 minutes. Okay. So in 3 minutes we won't be able to go into all the notation and arithmetics involved, but we can talk about the general conditions that mean field theory sort of assumes when we're applying coordinate Saint mean field variational inference. We are implicitly ignoring the dependency between the different parameters of this variational distribution. Instead of talking about very general eta, we'll separate it out to different variational parameters that are relevant for theta, for beta, and for z, all of the components of our actual latent variables. And we would assume that the distribution of theta d given theta d hat is completely independent of any of the other theta d's given their theta d hats, nor is it dependent on the betas and the z's. Under these conditions, the good news is that the distributions of each variable given its variational parameter have a very distinct form. It's proportional to an exponent in a particular expected log likelihood. And even more helpful is the fact that the distributions end up coming from the same family of distributions that we started with. So the variational distribution for theta would also come from a Dirichlet distribution. The betas would come up from a beta distribution, and so on. And with this, just to promise to point you to a wonderful review and tutorial by my departmental colleague Dave Bly, that really spells out a lot of the notations and definitions in a way that you can actually spend time with, and give a shout out to the actual application that we have coming out in August in the American Journal of Human Genetics that expands the model from just looking at the present, at the betas at the present, and talking about betas all throughout histories that offer a more expressive model that gives new insight about ancient DNA. Thanks. Yeah.
So you talked about how you can either use a direct approach using TensorFlow or using Cabbage. In what scenarios would you choose one over the other?
So some of it is development time versus complexity of the space. So if you have a natural, so if you're able to make the mean field assumption of independence of the variance given, independence of the parameters given their variational parameters, that's great. That gives you a more direct way to get your own update equations, and this would converge faster. If you don't, you can come up with a more approximate solution. It's that trusts the way the space is constructed, but you're optimizing in a huge space and assuming that your gradient optimization would actually give you the right solution without many assumptions. Yep.
So how much better do you do, I mean, if you consider the ancestry versus the model without considering the ancestry?
Thank you for asking. So how much better do you do? The y-axis is error. The x-axis is time of the simulated data of two populations where samples are from admixed individuals at different points in time, assuming we had ancient DNA from multiple sites that are carbon-14 dated from multiple points in history. Of course, the more time had elapsed, the more powerful our dynamic structure model is compared to the great implementation of structure admixture. This is making some parametric assumptions, but it doesn't matter really, and you can try different scenarios and still see significant advantage in simulated data. But simulated data is nice. What's perhaps more interesting is what happens when you work with real data. This is data by Lazaridis et al. From a couple of years back that included ancient samples as well as modern samples, because ancient samples are now accessible but are hard to come by. So the typical study that you'd see has many more modern samples that are a dime a dozen than ancient samples, which causes the training of the model to be very much driven by the modern samples. Now, it turns out that once you model the temporal semantics, you implicitly cause more ancient samples to be more pure. So in this case, these ancient samples are these European farmers are almost exclusively with the light green ancestry, as opposed to the more mixed bag of ancestries that come up from the standard analysis that includes lots of modern samples for training. What this does is helps interpretability of the data. When you actually look at the modern samples, remembering our color code, well, now we have a much better sense of, well, what is the modern day manifestation, or what are the modern day descendants of these early European farmers in the modern samples, something that's harder to interpret without the temporal model. Okay?
Thank you for this answer to my question.
Always have invited questions. And of course, since this is a tutorial, feel free as homework to figure out the variational inference model for this kind of temporal model, and answers are available in August. Thank you.